Welcome everyone to the second session of the Maddie Revolts Vision Centre of Research Excellence in Bone Marrow Biology expert webinar series. My name is Amy Coote and I'm the CEO at Maddie Revolts Vision. I would like to acknowledge the traditional custodians of the land on which we're gathered today. I'm joining you from the lands of the Wurundjeri people of the Kulin Nation. I'd like to pay my respects to their elders, past and present, and also acknowledge any Aboriginal or Torres Strait Islanders present with us today. As many of you know, Maddie's Vision is the only organisation in Australia driving bone marrow failure syndrome research, advocacy, education, and support for patients and families. We are incredibly proud to have established the Australian first Centre of Research Excellence in Bone Marrow Biology, and I'm very happy to welcome many executive members of our CRE here today. We now have multidisciplinary collaborations in place with 24 leading medical research institutes, hospitals and universities around Australia, and including the National Institutes of Health and Oxford University. We bring together the bone marrow failure syndrome scientific community to create opportunities for collaboration and innovation that otherwise would not have been available. We're the only source of bone marrow failure syndrome educational programs for both the scientific and patient and family communities. In addition to the session today, we'll be delivering another 2 expert webinars throughout 2024, with the next one planned for the 12th of September, featuring Professor Mary Armanios and Professor Tracy Bryan, presenting on telomere-mediated bone marrow failure. Additionally, planning is underway for our 4th National Symposium on Bone Marrow Failure Syndromes, which will take place from the 23rd to the 25th of May 2025 at the Victorian Comprehensive Cancer Centre. So please do save the date. We are absolutely delighted to have Professor Alan Warren as our expert speaker for the second of this webinar series. Following Alan's presentation, we'll have a 15-minute Q&A, so please do ensure you stay around for what will be a great opportunity for discussion. Before we do commence, I would like to acknowledge our partner Novartis for their generosity in sponsoring the 2024 CRE Expert Webinar Series. Novartis have become a valued partner to Maddie's Vision, and we're collaborating on a number of initiatives, including educational programs and patient and family engagement. Maddie's Vision relies on the support of partners, philanthropy, and the community to deliver on our mission, and we are so very grateful for their support. I now have the pleasure of introducing Professor David Ritchie, the Director of our Centre of Research Excellence, Chair of our Scientific Advisory Committee, Board Member of Maddie Revolts Vision, and highly valued source of support, guidance, and expertise to myself and the Maddie's Vision team. Over to you, David.
That's great, Amy. Thanks very much, and really magnificent to see all the work MRV is doing and continuing to build on. And of course, I'd also like to thank Novartis for making these sessions possible. So, as we've already kicked off in 2024, we've got a fantastic series of international speakers, and that is particularly compounded today by welcoming Professor Alan Warren, who is our expert speaker in this of the second of the webinar series. And as Amy has outlined, we'll have a Q&A capacity at the end of his presentation. Alan's a Professor of Haematology at the University of Cambridge in the United Kingdom, and we're really grateful for him giving up his time to share his insights with us today. He'll be discussing how new technologies are providing insights into the molecular basis of ribosomopathies, including Shwachman-Diamond syndrome, and really in keeping with Alan's forward thinking, how learning from that particular condition might allow us to learn about stem cell fitness and the development of blood cancers related to bone marrow failure syndromes, and how we might improve on the risk stratification and the personalised medicine approaches to SDS and inherited bone marrow failure syndromes more generally. So look, it's really with great pleasure and delight that I welcome Alan to present to us, and the floor is yours, Alan. Thanks very much.
So thanks very much for the invitation to speak today. It's a great pleasure. So I'm going to, as David's just said, I'm going to talk to you today about Shwachman-Diamond syndrome. And for those of you who are not aware, so there's a recent editorial in The Lancet which suggests that there's over 7,000 rare diseases affecting, maybe 300 million people worldwide. So rare disease is a very substantial disease burden. And one example of this is Shwachman-Diamond syndrome, which is a rare bone marrow failure disorder. It's a recessive disorder, and with a prevalence of about 1 in 200,000 live births. It presents in infancy, and patients may present with low neutrophil counts, sometimes complete marrow aplasia, and failure to thrive and poor growth. And significantly, about 30% of these patients go on to develop a blood cancer, typically myelodysplastic syndrome or acute myeloid leukemia in the 4th decade of life. So over 30% in the 4th decade, and there's currently no disease-modifying therapy. So there's a desperate need really to understand more about the biology of this condition to try to more effectively treat patients and improve their quality of life. Why did I get into Shwachman-Diamond syndrome in the first place? Well, when I set up the MDS clinic back in 2003 in Cambridge, the gene hadn't been identified at the time, and I thought perhaps because there was such a high proclivity to develop blood cancers, by understanding more about the disease, we might learn more about the pathogenesis of more common blood cancers. And actually, around this time, I had a conversation with the mother of a child with SDS, and that really stimulated my motivation to get involved in understanding more about SDS. So where does the name come from? So Christian Nazelov published the first report of the combination of pancreatic insufficiency and neutropenia, but because it was published in a French journal, it doesn't receive quite as much attention. So the eponymous designation Shwachman-Diamond comes from the two pediatricians, Harry Shwachman and Louis Diamond, who were working in Boston, and then Martin Bodine, who was working at Great Ormond Street, and they published a case report in 1964. The first one was Nazelov in 61. Although it's rare, if we look at the genetic distribution of congenital neutropenias in Europe, it's actually the second most common form of congenital neutropenia, accounts for about 21% of patients in the European register. This is data from Karl Velt. And we're still, this is the series from Coleman Lindsley, where he's looking at outcomes in young adults after transplant for myelodysplastic syndrome. So these are patients under the age of 40. As you can see here, so there was an unrecognized subset of these patients, about 4%, who had undiagnosed Shwachman-Diamond syndrome. And the point is they're doing really badly despite transplant. So, I'm still seeing patients in the clinic, presenting at the age of, in the 3rd, 4th decade of life, who, with undiagnosed Shwachman-Diamond syndrome. So we need to...
Alan, if I might just interrupt for a sec, do you think, is it possible to go to slide presentation mode? Because some of the text, it might be a bit easier to read. Yeah, that's perfect. Yeah. Thanks very much. Sorry about the interruption.
Okay. Yes, there's a desperate need, unmet need to try and understand more about the biology of this condition to better risk stratify patients and try and develop new forms of treatment. So that's what we've been doing essentially for the last sort of 20 years or so. So the SDS is predominantly caused by mutations in a single gene, SBDS. This accounts for more than 90% of cases of Shwachman-Diamond syndrome. The gene was identified by the team of Joanna Rommins based in Toronto back in, the paper was published at the end of 2002. And this is the structure of the SBDS protein, which we solved using both X-ray crystallography and NMR spectroscopy. And so it's a recessive disease, most commonly caused by a stop mutation in one allele and a donor splice site mutation on the other, but there's a range of other missense mutations, which were very informative for us in trying to understand the role of the SPDS protein. So really what we've been trying to do over the last few years is understand, address these key questions. What's the function, the basic function of the SPDS protein? How does the underlying genetic defect, which as I'll show is a defect in ribosome assembly, how does this promote the increased propensity to blood cancer? And can we harness these insights to risk stratify patients better and identify new targets for treatment? So I'll take these in turn. So first of all, because the SPDS protein is highly conserved in evolution, it was possible to use the power of yeast genetics to try to understand the role of the SPDS protein. So we took yeast cells which were completely lacking SPDS, and obviously they grew very poorly, but having scraped the cells off the plate and put them into liquid culture, after a few days, the cultures became turbid, and having worried that I'd contaminated the cultures, and repeating this multiple times, it became obvious, the penny finally dropped, that these cells were developing probably suppressor mutations, which allowed them to bypass the absence of the SPS protein. And just to cut a long story short, we identified mutations in a single protein, this EIF6, which stands for eukaryotic initiation factor 6, although it's actually it's more of an anti-association factor, as I'll show you in a minute. But all these mutations lined up on one face of the protein. We didn't know at the time, but we speculated that this might be the surface that interacts with the ribosome. And so the idea is that all of these missense mutations reduce the binding of EIF6 to the maturing large ribosomal subunit, and that's turned out to be the case. So these mutations are able to bypass the absence of SPDS by restoring ribosome homeostasis. So it suggested to us that SPDS was somehow involved in releasing EIF6, perhaps from the surface of the ribosome. Just to summarize quite a lot of work, we've sort of elaborated this model for the function of SPDS. So it cooperates. It's a cofactor for a GTPase, which is called elongation factor like 1. And together, these two proteins release EIF6 from the surface of the maturing large ribosomal subunit, so-called pre-60S subunit. And this is all happening in the cytoplasm. And the EIF6 is then released and is recycled back into the nucleus. It's essential to release EIF6 because it forms a steric barrier to the joining of the large and the small subunits. So obviously you need to join the large and small subunit to form an actively translating ribosome. And the reason is that EIF6 is sitting on the surface of the large subunit that interacts, that binds to the 40S. So it's absolutely essential to release EIF6 to allow for ribosomal subunit joining. And the problem in SPDS, in Shwachman-Diamond syndrome, which is caused by deficiency of SPDS predominantly, although we also see mutations in EFL, one is that EIF6 is retained on the surface of these cytoplasmic particles, and this impairs subunit joining and reduces, it has quite a dramatic effect on cellular protein synthesis, which is significantly reduced. More recently, we've gone on to show that SPS and EFL1 are more general EIF6 release factors, so they're not just involved in the biogenesis of the nascent 60S particle, but they're also involved in recycling subunits back into active translation, potentially following every round of translation. So once the ribosome reaches the stop codon, the small and the large subunits are dissociated, and there's the potential for EIF6 to again rebind to these free 60S particles. And but this we were able to show that there's a population of ribosomes in the cell which are in fact bound to EIF6, mature 60S particles. So there's kind of a sort of pool of ribosomes, which can then be actively re-engaged in active translation through the action of SPDS and EFL1. And recent in situ tomography, EM tomography images have shown that indeed there is a population of ribosomes in the cytoplasm of cells which is bound to EIF6. So a sort of functionally inactive pool, which must be re-engaged through the action of these two proteins. So the role of SPDS is much more pervasive, and that probably accounts for the significant defect in protein synthesis that we see in cells from patients. So this is the wild type, and here are three cell lines from patients. So we see a dramatically reduced rate of protein synthesis using the OP puromycin assay. Oops. So to better understand the role of the SPDS protein, the lab was based in the MRC Laboratory of Molecular Biology at around the time when we were undergoing this so-called revolution in single particle cryo-electron microscopy. So what we're able to do is to take ribosomes decorated with, large ribosomal subunits decorated with SPDS, EFL1 and EIF6, and try and understand how these factors interact with the ribosome. And we're able to access this enormous machine, which is called the Titan Cryos. It's enormous. It's 3 meters tall and weighs a ton, which is why it's called the Titan. And we're able to access these new direct electron detectors, which improves the signal to noise ratio dramatically and allows us to, as I'll show you shortly, to get true atomic resolution. So what we're able to show with this is that the SPDS protein interacts with a key functional site. This is the peptidyl transferase center of the ribosome. Here's the SPDS protein. This is the inter-subunit face. It's the face of the large subunit that binds to the 40S. So here's SPDS and here's EIF6, but there's no direct interaction between these two factors. And the SPDS is sitting in the exit tunnel of the ribosome. So it's blocking the ability of the ribosome to engage in translation. And our model is that SPDS together with the EFL1 is acting as a sort of quality control step. So if this nascent ribosome particle is formed properly, then SPDS engages with the surface and then promotes the release of EIF6 to allow to license, if you like, to license the entry of these large subunits into active translation. So how does it work if how is EIF6 released if SPDS doesn't directly interact? We're able to generate images, cryo-EM images of SPDS together with the GTPase, which is directly contacting EIF6. We'll just move this around slightly. And you can see there's a very intimate contact between the GTPase and EIF6. So the SPDS is acting as a sort of allosteric regulator, we think, and it's the EFL1, the GTPase, that's doing the heavy lifting. So we think there's a release of EIF6, and then EFL1 binds to the ribosomal RNA and then hydrolyzes upon hydrolysis of GTP, SPDS, and EFL1 is then released, but there's still more work to be done to understand exactly how SPDS EFL1 promote the release of EIF6 from the surface. But anyway, the fundamental role in all cells is the SPDS EFL1 release EIF6 to license the entry of these mature 60S subunits into active translation. And of course, we, as I said, STS is predominantly caused by mutations in this SPDS protein, but we also have a subset of patients with mutations in elongation factor like 1. And again, we see profound effects on these ribosome profiles and a significant reduction in protein synthesis in cells derived from these patients. And subsequently, the group of Yigal, Dror, and also Inderjit Dokal at the London Hospital identified patients with mutations in another factor. This is a J-domain protein, which is called DNA JC21. So, yeah, and then more recently, mutations in this component of the signal recognition particle, SRP54. They have an SDS-like phenotype, although these patients are autosomal dominant. The DNA JC21 are also autosomal recessive. So multiple components of this cytoplasmic ribosome maturation pathway are targeted by disease mutations causing, and we can debate whether this is SDS or SDS-like, but certainly SVDS and EFR1 mutations, they phenocopy the sort of classic SDS phenotype with pancreatic insufficiency, bone marrow failure, and skeletal problems. And the DNA JC21 also looks very similar. So we over the years, we've been trying to understand exactly when these factors are working, and they're still work in progress. We've been using cryo-EM to try to understand the entire, the maturation process, at least in the cytoplasm in some detail. I'll just show you this movie. This is based on data from yeast, but it illustrates the sort of dynamic changes which are happening during maturation of the ribosome in the cytoplasm. These are the final steps in the process. So this is most of this is RNA. This is the ribosomal RNA, which is decorated by a variety of different ribosomal assembly factors and ribosomal proteins. This is the intersubunit face, which contains, as you can see, a lot of protein, which is blocking ribosome assembly. So all of this stuff has to be released in a sort of ordered and regulated manner. And what I want to illustrate here, so this is recruitment of UL16, which is one of the ribosomal proteins, which is mutated in T-cell leukemias. And this promotes quite a substantial movement in the ribosomal RNA. This is one of the ribosomal RNA helices. And then this long factor, NMD3, is then released from the intersubunit interface through the action of a GTPase, which is called LSG1. So the details aren't important, but it's just essentially what's happening here is we're liberating all these factors from the functional surface of the ribosome. Then this is the SVDS protein that comes along and interrogates the conformation of the ribosome, then recruits EFL1, which, if the conformation of this particle is correct, then drives release of EIF6. And we're generating similar movies for the human ribosome, and hopefully we'll be able to publish that very soon. And because we're so frustrated at our inability to visualize this in sufficient detail, Alex Fye in my lab has developed new tools which now allows them to interrogate human ribosome structure and function at very high resolution. So a resolution of about 1.3 angstroms. And what does that mean? It means we can see actual hydrogen atoms. We can see all the solvent molecules. We can see water, magnesium, potassium, as well as all the base modifications such as the methylation and pseudo-uridylation. And I think this is going to be a game changer in terms of structure-based drug design, as well as trying to understand the mechanistic details of how we make these machines, these nanomachines that are responsible for making all the protein in our cells. So I want to spend a bit of time now changing topic and thinking about this question, which has become known as Damashek's riddle. How do stem and progenitor cells escape the fitness defect that's caused by a germline defect in ribosome assembly? And to address this question, the obvious idea would be that cells might restore fitness by mutating EIF6, similar to what we saw in yeast. And so we teamed up with Patrick Rivi, who had access to the French SDS cohort, and asked that question. So do patients with Shwachman-Diamond syndrome have somatic mutations in EIF6, which might in principle restore ribosome homeostasis? And of course, that turned out to be the case. So somatic EIF6 mutations turn out to be really frequent in Shwachman-Diamond syndrome. And in fact, many of these mutations, for example, this N106S mutation, were mutations that we found in yeast, so it actually completely replicates the findings in Saccharomyces cerevisiae. We also found, so this interstitial 20q deletion is it's been described for many years in patients with STS, but it also encompasses the EIF6 gene, but there's also a lot of other genes in this, in this deleted region. But we're able to show a patient with a reciprocal translocation, which takes out EIF6. So it does look as though the key gene in this interstitial 20q deletion is EIF6 itself. So we've got somatic mutations, deletions, and reciprocal translocation, all involving EIF6. So what are the functional consequences of these mutations? So we get able to show again using our high resolution structures that these mutations map to distinct sites of the EIF6 protein. So many of them cluster at the interface with the ribosome. This is a protein called UL14, which mediates the contact with the EIF6. And then some of the mutations lie quite distally, and these are mainly destabilizing and unfold the protein. And we're able to look at the functional consequences. So some of the mutations, they reduce the amount of the EIF6 quite significantly. The proteins are very unstable. But this N106S, the protein is expressed at similar levels to the wild type. But what we could show is that it rescues the SPDS deficient yeast cells, and biochemically, the N106S doesn't have the same affinity for these cytoplasmic 6DS particles. We see most of it in the free fraction compared to the control. So the effect of the N106S is to reduce the affinity of EIF6 for the ribosome. And some of the other mutations are simply reducing the stability. Just for fun, I'm going to show you this movie. So this is a molecular dynamics simulation, and we've got the wild type on the left here, and then the mutant on the right. So this is the mutation, the N106 to serine. And so we basically put this structure into a box and added a solvent molecule. And what we can see is that the water molecules in our mutant quite rapidly over the course of 500 nanoseconds are able to solvate, so they're able to enter and disrupt the interface between EIF6, which is shown here in yellow, and this ribosomal protein UL14, whereas the wild type protein sort of rock solid over the course of the 500 nanoseconds. So the consequence of introducing this mutation is to solvate the interface and disrupt the interaction of EIF6 with the surface of the subunit. We've taken this a bit further more recently using NMR spectroscopy, and what we find is that despite this mutation sitting right at the interface, the introduction of a serine into this position has quite a dramatic effect on the protein, even at distal sites. This is bar-coded depending on the extent of the perturbation. So a lot of the perturbation is here, as you might expect, close to the mutation, but we also see disruption of the fold of the protein at quite distal sites. So these mutations, we're still trying to understand more about precisely how they work using these new techniques. This work is still ongoing. So what was really amazing was that if we take, and we've done this with several of the somatic mutations, if we add them back to our genetic models, this is a fly model, so this is the wild type, and here are SPDS deficient flies, which die quite early during larval development. But if we add back this N106S EIF6 as a transgene, we completely restore the viability and fertility of these flies. And also, if we reduce the dose of EIF6, we can also at least partially rescue the viability of these of this animal. We've also repeated, this was a transgenic overexpression, but we've also engineered flies having a stop mutation in the SPDS protein in the genome, a single genomic copy. And again, we can rescue with a single genomic copy of the EIF6 mutation. So it's a very robust assay, which shows that these somatic mutations have the ability to rescue the germline SPDS mutation fully. So this leads to the idea of somatic genetic rescue, and our model nicely explains a number of the chromosomal abnormalities which we see in patients with Shwachman-Diamond syndrome. So one of the most common mutations is isochromosome 7, which increases the amount of the wild-type SPDS. As I said, most patients with Shwachman-Diamond syndrome are compound heterozygous for a stop and the donor splice site mutation. So this isochromosome 7 duplicates the donor splice site, so we get more SPDS protein. And we've observed amplification of chromosome 15, which increases the amount of wild-type EFL1. Other common cytogenetic abnormalities are interstitial 20q deletion, which reduces the amount of EIF6. And we've now shown that EIF6 missense mutations, somatic mutations are very common in STS. And so again, all of these mutations are restoring ribosome homeostasis by increasing the amount of the critical factors for release of EIF6, or reducing the amount of EIF6, and thereby allowing restoration of ribosomal subunit joining and restoration of protein synthesis. But we wanted to try and understand this in a bit more detail, so and look at the effect of these somatic variants on clonal dynamics. And so in order to do this, we've taken advantage of this new technology that allows us to map mutations at the level of single hematopoietic stem cells. And so what we're doing here is tracking spontaneously acquired single nucleotide variants that occur in hematopoietic stem cells. These mutations are occurring linearly with time, so we're all acquiring about 15 mutations a year per genome. And by comparing common shared mutations, it allows us to draw these family trees. And so the advantage of this technique, it's expensive, but the advantage is that we can generate a lot of information, in particular the timing of mutation acquisition. It gives us information on total mutation burden at a single clonal level, also mutational signatures, the degree of clonal expansion caused by the acquisition of specific variants, and comparing single nucleotide variants across different clones. So this is an example of a phylogenetic tree from a normal 29-year-old, which shows it's highly polyclonal. But what I'll show you is an example of a patient with Shwachman-Diamond syndrome, where there's very marked loss of clonal diversity. So this phylogenetic tree looks more like what you would expect to see in a 70-year-old patient, whereas this young adult was 22 at the time of first sampling, 24 when we took the second sample. And so about half of his hematopoiesis is made up of a very restricted number of stem cells. And what you can see is that there are these large expansions shown here, for example. Each of these points is a single colony that we've sampled, and because mutations are occurring, are accumulating linearly, it allows us to map these mutations in time. And what we notice is that each stem cell takes a different route out of the problem. So we've got mutually exclusive acquired recurrent mutations, and the most common genes that were affected were P53, EIF6, and then we also identified mutations in these two ribosomal proteins, L5 and L22, as well as structural anomalies such as copy number variants in chromosome 7 and also in chromosome 15. And this was, we observed this phenomenon across, the 10 patients that we looked at. Loss of diversity, clonal diversity, acquisition of multiple independent but recurrent mutations in these genes. And I don't think this list is still not comprehensive, and I suspect we're still going to. Well, we have found additional genes, and there may be a lot more still to find. And the other advantage of this technology is that, as I said, we can go back in time and assess exactly when these mutations were acquired. And it was kind of surprising to me, at least, that many of these mutations, and so this patient had acquired the P53 and the EIF6 mutation in utero, and the clones are persisting for decades. And then the other piece of information we can derive is looking at the signatures and also the rate, the number of total number of mutations, the mutation burden these patients acquire at the and we can look at this at the level of single hematopoietic stem and progenitor cells. So, as I say, the number of mutations acquired in life is linear, about 15 per year per genome, and all the clones that we sequenced lie along this line, apart from this clone, which contained a biallelic TP53 mutation. And this was also characterized by the presence of numerous structural anomalies, and we can estimate the rate of expansion of this thing from acquisition of the P53 mutation to presenting with MDS in this patient. It took about 6 months, so very, very rapid time course, which in terms of thinking about how to manage these patients presents us with some considerable difficulties. What are the types of mutation? So what we found is the SBS blood and so-called SBS blood and SBS1, and these are C to T transitions predominantly, which is the common variant in blood. But the SBS1 is a characteristic of rapidly proliferating cells, and we see an increase in the proportion of cells in the patient, in this patient who went on to develop MDS. So we've got an increased enrichment of these SBS1 mutations, and our interpretation is this simply reflects the increase in the rapid outgrowth of this clone. So the somatic mutations are providing a substrate for clonal selection in the P53 mutated cancer evolution. So our model then is that in SDS we have convergent, what we call convergent somatic evolution. So all these somatic variants are rescuing the fitness of the cells by trying to bypass this so-called nucleolar stress pathway. So deficiency of SPD-SEFR1 leads to reduced ribosome assembly. This stimulates nucleolar stress, which leads to the accumulation of P53, because this complex, the 5S RNP complex, binds to the E3 ligase, HDM2, and inactivates it. So we stabilize P53 in the SDS patients. So the convergent evolution leads to, firstly, increased dose of SBS or EFR1, reduces the dose of EIF6 potentially. Thirdly, the mutations target components of this 5S RNP particle. This still needs to be tested really for the L22, although L5, we think that's probably what's happening. And then fourthly, the mutations can target P53 directly, thereby enhancing clonal fitness, but setting up the possibility of acquiring biallelic P53 mutations and the progression to malignancy. So this is a more simplified version of this model for progression to blood cancer. Patients with SDS, with the germline ribosome defect, this is a severe fitness defect, which is bypassed by these, what we might describe as either maladaptive or adaptive somatic genetic mutations. EIF6 mutations are adaptive as they restore ribosome homeostasis, whereas the P53 mutation sets up the possibility of biallelic P53 mutation acquisition and transformation. And what was perhaps a surprise is that just a single hit in P53, these clones sit around, they have a fitness advantage, but they don't progress to malignancy. So one of the lessons is that if we see a range of, as we do, up to 10% in some cases of clones carry single heterozygous P53 mutations, it's not an indication to transplant unless we note that there's a huge increase in the variant allele frequency. And so going forwards, is it possible? We're wondering, could we try to remove these so-called maladaptive clones? Could we develop strategies to potentially enhance the expansion of so-called adaptive mutations, such as the CIF6 mutation? And can we use this information to better stratify patients? So if patients presenting with a biallelic P53 mutation, it would be because we know that these grow out so quickly and progress to malignancy. This is probably an indication to transplant if there's sufficient time, if we can detect these biallelic clones. But the question is, how do we go about doing that in a robust manner? And then finally, the identification of these EIF6 suppressor mutations sets up the possibility of trying to mimic the effect of the suppressor mutations with small molecules. So, having established the molecular basis of the disease, we've shown genetic proof of principle that these EIF6 mutations can completely rescue the viability of the flies. So the task that we've set ourselves is to try to identify small molecules that can mimic the effect of the suppressor mutations and disrupt the interaction of EIF6 with the ribosome. And I'll hopefully be able to report on progress in this over the next few years. So to summarize, SDS is a ribosomopathy that's caused by defective maturation of the large subunit. Somatic mutations provide a substrate for clonal selection, and although they offset the deleterious effects of the germline ribosomopathy, they provide an opportunity for P53 mutated cancer evolution. This sets up the rationale for trying to develop EIF6 suppressor mimics potentially as therapeutics. Now there are opportunities from what we've now learned to improve precision medicine through improved marrow surveillance and possibly early intervention. I'd like to thank the patient groups in particular. So we've tried to set up this multidisciplinary clinic here in Cambridge, and this was the first clinic that we set up. We've got there's a study which has just started by Genomics England to look at 100,000 patients over the next year and screen for over 200 disease mutations, including a number of bone marrow failure conditions. This is an exciting initiative. It includes all the SDS genes. We've set up this national charity which provides advice and support to patients and also to our lab, which we're very grateful for. And many of these rare disease charities have teamed up together to obtain lottery funding, particularly for psychological support for patients. So the idea is they're better together and better able to get resource. And this is the team. This is the lab. And thanks to the funding bodies, and thanks for your attention. Stop there.
Thanks very much, Alan. Perhaps Yeah, thanks. I'll share your screen there. So we do have a Q&A session for the next 10 minutes or so. I encourage people to put their questions in the chat, please. And I think, Alan, you should be able to see the chat if you want to read them yourself, but I can read them out if it's easier. So Parvati Venugopal, one of our esteemed researchers, and she's asked, she said, well, obviously, thanks for your great talk, which was an excellent talk. She's really specifically asking about a chromosome 7 abnormality in STS patient, and what were the germline SBDS variants in that patient? Is it possible that one of the variants was less deleterious than the other? Would you say that it was a maladaptive event, i.e. a monosomy 7 occurrence?
So, so I think if I remember correctly, all of these patients were compound heterozygous for the stop and the donor splice site mutations. So, so what happens in these chromosome 7 variants is that there's loss of heterozygosity, so it results in a duplication of the allele that carries the donor splice site mutation, and so this results in an increase of wild type SBDS protein expression. So I'd suggest that this was an adaptive. It's an adaptive event, as opposed to a monosomy 7.
Right, and so along these same lines, Lucy Fox, who you know, also congratulates you on your talk and the graphics associated with it. And you may be able to read her question there, but basically, you know, the $64 million clinical question, which is, you know, should we go about some form of routine monitoring for maladaptive changes? And alternatively, what about adaptive changes? Is there a way to potentially give patients good news by serial monitoring?
Yeah, so yes, it's a great question. So we've largely stopped doing bone marrows because what we suggest is that patients should have a full blood count once every 3 or 4 months, and we combine that with screening for somatic variants once every 6 months at the moment, because we don't know the natural history, how frequently patients might acquire these mutations going forward. So at the moment, 6 monthly screening for somatic variants, and also microarray, so array CGH analysis to look for structural variants. So we're suggesting that we do that at least 6 monthly, maybe once a year. But the key thing is to keep repeating the blood counts more frequently, because we had one patient who, unfortunately, we did the marrow and then we blew up with AML within. I mean, he presented within 4 months, and we, it was a P53 clone in the leukemic clone that we hadn't detected using these methods. So I think there's a limitation with the current clinical, the depth of sequencing, and so what we're trying to do is to enhance the depth of sequencing and try and find these rare clones. I mean, the biggest challenge, obviously, is trying to find these biallelic P53 clones, and we're looking at perhaps using some of these platforms to try and find them. But how many clones do you screen, and how often do you do it? That's something that we need to decide as a community. We've really struggled with the bone marrows. Most of our patients have to have general anaesthetics, and even the quality of the sample has not been really improved by having the anaesthetics. So we've tended to go for these analysis on peripheral blood. We're not really seeing differences in the variant allele frequencies, significant differences comparing bone marrow and blood. So we're sort of fairly confident we're not missing anything critical, and we are now screening for EIF6 mutations, L22, L5, both locally, and we're trying to get this commissioned nationally as well. And also, we can try and we're working with Akiko and Coleman, so they're screening their cohort of 400 patients. So we'll see how common these L22 and L5 mutations are. But as I say, I suspect we've only found the most common ones, and there's still more to find. I mean, the most common route to malignancies is the acquisition of these biallelic P53 mutations, but it's probably not the only route, but that's what we're seeing most commonly.
So in that same vein, and with a slight clinical extension as well, Paz Barbaro has asked about the ELF6 somatic variants and other adaptive changes, and whether that's completely reverses the phenotypic risk of MDS and AML, and can it be extended to clinical good news? You don't have to come to clinic anymore. And then, sort of as a related question, what about the use of growth factors in patients with cytopenias?
So, so unfortunately, finding large EIF6 clones that isn't entirely good news. It does, based on data, it's largely from Italy. We've looked at this in more detail, and certainly from our clinical experience, most of the patients with the large 20q clones are doing reasonably well. They have good counts, but there are still reports of patients developing leukemia. So obviously there's still the possibility of a p53 mutation blowing up from an independent clone, unfortunately. So I guess if you could hit, possibly with a drug very early on, we know these mutations are arising in utero. So could we sort of try and protect the cells by reducing this ribosomal stress early enough to try and prevent acquisition of p53 at all, or perhaps reduce the risk of getting the second hit in p53. There's one patient that we reported in the paper last year, the one from Singapore, who the bone marrow cellularity recovered and the blood counts recovered, and that was correlated with a rise in the EIF6 containing clone. So it does seem to restore hematopoiesis in some cases, but it's not going to be fully protective, unfortunately, from getting AML in another clone. And experience with PEG GCSF. So we haven't really used a lot of PEG GCSF in SDS, but it's obviously a very effective therapy in general. Most of the adult patients aren't they're not GCSF dependent unless they develop a severe infection. So we don't have anybody at the moment who's on chronic GCSF treatment.
Yeah, so I was going to ask a question similar to the one that Ashley's posed, so we'll ask his one instead, which is there a difference in the approach about the clinical surveillance and or the risk for the patients who perhaps have been identified as young adults or children who now are older adults, or those who indeed present as, shall we say, more middle-aged? Is there a different approach in those age cohorts?
Yeah, so obviously the number of somatic mutations is increasing linearly with age. So as you get older, particularly in the over 30s, the risk of MDS leukemia appears to be much higher. And we've already lost 2 patients in the last 5 years, which is just unbearable, really. So we've been quite we've been insisting on having these 3 to 4 monthly blood counts and really going overboard perhaps on the somatic variant screening, but if having to go to funerals of patients is not something I want to do routinely. So
Before we get on to Lucy's second question, I'm just going to extend a question that along the same theme as Ashley has posed. So at our site, we've had a lady walk through the door for the very first time in our service, and indeed to any haematology service, in the mid 50s with p53 mutated AML, who did not unknown Shwachman-Diamond syndrome and did systemically poorly with chemotherapy, as you might guess, but did go into remission, but only just survived the therapy. You mentioned through your talk about the relative risks of p53 and the biallelic mutations and the trigger for transplant. In most instances in transplantation for de novo AML, we get a bit pessimistic about the ability of transplantation to alter p53 mutated AML. What's your view about that?
So I think that's true. There may be a window of opportunity. So from acquiring the biallelic p53 before you develop all the downstream structural anomalies. So Akiko has a case where she transplanted the patient early on, just following the acquisition of the biallelic P53 mutation, and that patient, as far as I know, seems to be doing well. So perhaps there's a window of opportunity within that very small interval. I mean, as I say, the two patients that we've had recently, the P3 clones blown up and caused problems within 6 months. So, I mean, it just seems like an impossible challenge. You know, they become chemo-resistant, the two patients. I mean, the second one we couldn't get into remission, but actually he had a sort of biphenotypic. It was really odd. It had features of ALL as well as myeloid. So we really struggled to get him into remission, and he didn't recover his counts. And then the other, the lady, we transplanted, but she relapsed very quickly. But they both had, large numbers of structural variants by the time we came to even think about treating them. So yeah.
So as the sort of tapestry of these conditions becomes increasingly complicated and more threads of molecular lesions lead to similar sorts of phenotypic descriptions, Lucy's posed this question, which is, do the sort of more newly defined SDS-like lesions, such as the ones you've outlined, do they have an equal risk of evolving myeloid malignancies? And is there any alternative pathway to treatment for those subclass diseases, if you like?
Yeah. So the EFL1 is still very rare, and so I don't have any data on progression to MDS AML, but the DNA JC21. So we've got one patient who seems to be moving. He's now got biallelic P53 mutations, and the reports from North America, from the Canadian registry, that several of their patients have progressed, but whether the rates are similar, I don't think there's enough data yet, but it certainly seems to be happening, and it looks like it's P53 mediated. We've had a chance to look at one patient only so far, using this new technology and drawing phylogenies, and again, they have the same set of mutations, L22, L5, but not EIF6 and P53. So it's quite interesting.
So we're almost at time. I might just take Chair's prerogative to ask the final question, and thanks to those people who posted questions. Excellent opportunity for conversation. So Alan, as the genotyping extends and becomes perhaps clearer, or even potentially even muddier as more and more aberrations are identified, do you feel that that the incidence of SDS or related conditions is much higher than we appreciate? And is there a need, and indeed is there a mechanism by which these forms of inherited bone marrow failure syndromes become a public health issue, trying to identify cases who might not present to haematologists, or indeed they might and still not be recognised, but maybe presented to the general paediatricians, the general, the dietitians, the respiratory doctors, and so on? Is there some public health initiative that's required for these rarer conditions?
Yeah, I think it's becoming an increasing problem. So we have several patients who've presented through the genetics department, gastroenterology, as you say, or presenting with frank AML and getting to transplant even without without recognizing they have SDS. So the initiative in the UK is to screen all newborns, and as I said, there's this 100. The NHS England is just beginning this pilot study on 100,000 patients, and was involved in the discussions with them about which genes to incorporate. So we've got many inherited bone marrow failure syndromes, but obviously the ones that that's amenable to some sort of intervention within the first 6 months of life. So all the SDS genes are in there. I mean, we've got patients just coming to clinic with neutropenia, and we've been screening them using the various genetic panels, and surprise, we've turned up a lot of patients with inherited bone marrow failure disorders. So yeah, it's very underdiagnosed at the moment. Yeah, okay. I think it's a public health issue.
Yeah, thanks, Alan. So So that is time, and I really would like to very sincerely thank you for your time, Alan. We appreciate you're busy and appreciate you have a lot of requests to talk. So the audience will be very grateful for the time that you've been able to spend with us. Just for those people who are participating in today's webinar, the crew in the MRV office will be sending you out a survey regarding your views on the value of this these types of sessions, and we know that you will score them extremely highly, as because they're incredibly insightful and really cutting edge. We hope to see you all back and invite your friends as well to future editions of our webinar, and of course we thank our sponsor Novartis for their support through this, and thank you again, Alan, and we wish you all the best and hope to see you again soon. Good night, everybody. Thanks very much.
Thanks a lot. Nice to see you all. Cheers.
Thanks, Alan.
See you in Paris.